Bismillahirrahmanirrahim. Assalamu alaikum. I am Anas Mahmood and my friend Saim Khan will tell you about the fluorescence and phosphorescence. First of all, you have to be familiar with the term photoluminescence. Photoluminescence is a process in which a substance absorbs photons and then re-radiates photons. An excitation to a higher energy state and then a return to a lower energy state accompanied by the emission of a photon. Next one is forms of photoluminescence. There are two forms of photoluminescence, fluorescence and phosphorescence. First one is fluorescence. Fluorescence is the emission of electromagnetic radiation light by a substance that has absorbed radiation of a different wavelength. It is a short-lived emission and luminance stops immediately within 10 raised power minus 8 to 10 raised power minus 5 seconds after irradiation is discontinued. In this picture you can see the fluorescent minerals which emit visible light when exposed to ultraviolet light. Next one is phosphorescence. Phosphorescence is commonly defined as the delayed emission of light from a substance after exposure to and removal of the exciting radiation. The phenomenon of phosphorescence is a long-lived emission lasting from 10 raised power minus 3 seconds to the order of seconds. In this picture you can see the fluorescent bulb embedded in a block of acrylic. It requires no electricity and can glow for 3 hours. Now the comparison of fluorescence and phosphorescence on the basis of electronic energy transition. I have already told you about the terms fluorescence and phosphorescence, so we can easily compare these two terms. Fluorescence differs from phosphorescence in that the electronic energy transition that is responsible for fluorescence does not change in electron spin, which results in short-lived electrons, that is less than 10 raised power minus 5 seconds in the excited state of fluorescence. But in phosphorescence there is a change in electron spin which results in a longer lifetime of the excited state, that is from seconds to minutes. This picture shows how atoms emit light. Here you can see the 3 spots where we have indicated the 1, 2 and 3. Point 1 shows a collision with a moving particle excites the atom. Point 2 shows an electron jumping to a higher energy level, and the point 3 shows the electron falls back to its original energy level, releasing the extra energy in the form of a light photon. The electronic states of most organic molecules can be divided into singlet states and triplet states. Singlet state: electrons in the molecule are spin paired, and in triplet state one set of electron spins is unpaired. In these arrows shows the ground singlet state, excited singlet state and the excited triplet state. How fluorescence occurs? Fluorescence occurs when the molecule returns to the electronic ground state from the excited singlet state by the emission of a photon. If a molecule which absorbs UV radiation does not fluoresce, it means that it must have lost its energy some other way. These processes are called radiation-less energy or transfer of energy. And how phosphorescence occurs? Emission of triplet or singlet transitions can continue after initial irradiation. These discontinuous radiations are called phosphorescence. Now I'll tell you about the internal conversion, vibrational relaxation and the intersystem crossing. The first one is internal conversion. When the molecule returns to the electronic ground state, the excess energy is converted to vibrational energy. This is called internal conversion. Next one is vibrational relaxation. Excess vibrational energy is lost by collision with other molecules. It is called vibrational relaxation. And the last one is intersystem crossing. The spin of an excited electron can be reversed, leaving the molecule in an excited triplet state. This is called intersystem crossing. Next one is factors affecting fluorescence and phosphorescence. First factor is temperature. Quantum efficiency of fluorescence decreases with increasing temperature. As the temperature increases, the frequency of the collision increases, with increase the probability of deactivation by external conversion. Second effect is solvent effect. Solvents with lower viscosity have higher possibility of deactivation by external conversion. Fluorescence of a molecule decreases when its solvent contains heavy atoms such as carbon, tetrabromide and ethyl iodide. Orbital spin interaction result from an increase in the rate of triplet information, which decreases the possibility of fluorescence. Heavy atoms are usually incorporated into solvent to enhance the phosphorescence. The last one is effect of dissolved oxygen. The presence of dissolved oxygen often reduces the intensity of fluorescence in a solution. This effect may be the result of a photochemically induced oxidation of the fluorescing species. Now my friend Saim Khan will further elaborate this topic and tell you about the instrumentation and further about this topic. Thank you very much.
Now I'm going to tell you about the instrumentation that can help us detect the presence of fluorescence and phosphorescence. First of all, the instrument used for measuring the presence, that is, its intensity and wavelength distribution of fluorescence, is called fluorometer, while the instrument used for detection of phosphorescence is called phosphorimeter. The main components of fluorometer and phosphorimeter are light sources, cells and sample compartments, wavelength selectors, detectors. While there is also an extra feature for phosphorescence, where a sample is placed inside a cooled Dewar flask with liquid nitrogen so as to keep the temperature low. Fluorometers. There are two types of fluorometers, filter fluorometer and spectrophotometer. First of all, I am going to discuss about filter fluorometers. Filter fluorometers produce specific excitation and emission wavelengths by using optical filters. The filters block other wavelengths but transmit wavelengths relevant to the compound. The light passes through the sample to be made, and a certain wavelength is absorbed while a longer wavelength is emitted. The emitted light is made by a detector. By changing the optical filter, different substances can be made. As you can see in the diagram, a lamp is used to produce light which passes through the excitation filter so as to filter out a specific wavelength of light. Then this filtered light falls on the sample which absorbs it and emits certain wavelengths, which then passes through the emission filter. And the emission filter filters the specific wavelength to sample, and finally a light detector gives out the digital readout when these wavelengths falls on it. Next is the spectrophotometer. Spectrophotometer is an instrument which takes advantage of fluorescent properties of some compound in order to produce information regarding their concentration and chemical environment in a sample. A certain excitation wavelength is selected and the image is absorbed either at a single wavelength or at a scan is performed to record the intensity versus wavelength, also called an emission spectra. As you can see in the diagram, an arc lamp is used to produce light of certain wavelength, which goes on through a slit onto an excitation monochromator, which excites the wavelength, and after passing through another slit, it goes on to the sample and also to the reference sensor, whose duty is to compare the sample with a certain reference material. And after the wavelength falls on the sample, which goes on to the emission monochromator and onto the PMT, which gives the readouts. Now, phosphorimeter. Fluorescence can only be measured if the sample is solid or at liquid nitrogen temperature, that is negative 196 degrees centigrade. The basic instrumentation is similar to that of the fluorescence, apart from the sampling technique and the recording procedure. Sampling technique. In case of phosphorescence, ethanol is used as solvent for photomolecules with addition of small quantity of acid or base. On the other hand, a mixture of diethyl ether, isopentane and ethanol in a ratio of 5:5:2, commonly called EPA, in an excellent choice for non-polar components. Recording procedure. A mechanical shutter is used to execute recording of phosphorescence in order to cut off the incident radiation which ceases the fluorescence immediately, whereas the phosphorescence emission continues, which can help us to detect the presence of phosphorescence. As you can see in the diagram, a Dewar flask containing liquid nitrogen which has been placed with a sample tube. This liquid nitrogen is there so as that low temperature, that is of 196 degrees centigrade, can be achieved. And there's also an excitation shutter and emission shutter. The ray of light passes through the excitation shutter onto the sample and goes on to from the emission shutter to the detector, which gives the readouts, and hence the presence of phosphorescence can be detected and recorded. Now I'm going to discuss some of the applications of fluorescence and phosphorescence. First of all, visualization of specific submolecular structure. Fluorescence imaging can be used to visualize specific submolecular structure. As you can see in the first image shows microtubules stained with a fluorescent dye. The image was recorded in black and white. The second image shows nuclei stained with a fluorescent compound. This image was also recorded in black and white, what was obtained using excitation light of different wavelength than the microtubule image. In the third image, to obtain separate colors were assigned to the two previous images before combining them into one. Next, detection of biological molecules. Biological molecules can be tagged with a fluorescent chemical group called fluorophore by simple chemical reaction, and the fluorescence of tag enables sensitive and quantitative detection of molecules. Next is the application of phosphorescence and fluorescence in biochemistry and medicine. Fluorescence in the life sciences is used generally as a non-destructive way of tracking and analyzes biological molecule by means of the fluorescent emission at a specific frequency. As you can see in the diagram, endothelial cells under the microscope with three separate channels marking specific cellular components. Next is the determination of uh, atropine and cocaine. Atropine and cocaine and p-aminobenzoic acid at room temperature can be determined in urine by the phosphorimeter. Also, with the combination with the photochromatography, phosphorimeter with the combination with the thin layer chromatography has been used in separating three tobacco alkaloids, that is nicotine, nicotine and anabasine. These techniques help us to extract different components in certain samples. Other applications are determination of pesticides, uranium determination, visualization of fingerprints, and determination of aspirin in blood serum. That's all, and I hope we have been able to provide you some knowledge. Thank you very much.